Hello everyone. I hope all of you watching this video are staying at home and studying diligently. If you currently do not have school and don't have enough resources, you can look forward to my new series of slides. Let's get into today's lesson, part 1 of the topic of cellular functions, eukaryotic cells. Here is a diagram of a eukaryotic cell. A nucleus is present in every eukaryotic cell, but absent in prokaryotic cells. Eukaryotic cells always contain more than one chromosome. Instead of chromosomes, prokaryotic cells contain a plasmid. Mitochondria are present in eukaryotic cells, but not in prokaryotic cells. Animals and plants are eukaryotes while bacteria and archaea are prokaryotes. Let's move on to key idea A, cell organelles. The nucleus is the largest organelle and is enclosed within a nuclear envelope comprising of double membranes. The outer membrane is continuous with the endoplasmic reticulum. Within the nucleus is a semi-fluid matrix called nucleoplasm, where chromosomes are found. Most DNA within a cell is located inside the nucleus. Chromosomes are made up of DNA associated with proteins. Different species have different number of chromosomes. For example, in humans, there are 46 chromosomes in the nucleus of a diploid cell and 23 chromosomes in the nucleus of a haploid cell. Nucleoli are responsible for the synthesis of ribosomal ribonucleic acid. The nucleus contains the hereditary material of an organism. The DNA in the nucleus contains genes which control and direct activities of a cell by regulating protein synthesis. The nucleus divides during cell division, which is a part of the growth and development of multicellular organisms. The mitochondrion is bound by a double membrane. The outer membrane is smooth and continuous while the inner membrane is extensively folded to form cristae. The membranes are separated by the intermembrane space. The mitochondrial matrix contains circular DNA and 70S ribosomes. Mitochondrion is the site for cellular respiration to produce adenosine triphosphate. The rough endoplasmic reticulum originates from the outer membrane of the nuclear envelope and consists of a network of flattened membrane-bound sacs called cisternae. Ribosomes are attached on the outer surface of the membrane. The rough endoplasmic reticulum is the site for protein synthesis by the attached ribosomes and subsequent transport to the next cellular location. The smooth endoplasmic reticulum consists of a series of interconnected tubules. The smooth endoplasmic reticulum is the site of a variety of metabolic processes, namely, lipid synthesis, the metabolism of carbohydrates, the detoxification of drugs and the storage of calcium ions required for muscle contraction. The Golgi apparatus, also known as the Golgi body, consists of a stack of membrane-bound sacs called cisternae and Golgi vesicles. Cisternae are continually being formed at the cis base of the Golgi body. This occurs when vesicles that bud from the endoplasmic reticulum fuse with the Golgi apparatus and add their content into the lumen of the Golgi body. The other end of the Golgi apparatus is called the transphase. It breaks down continually as vesicles pinch off to transport the biomolecules elsewhere. At the Golgi apparatus, proteins and other biomolecules are sorted and packaged before transportation to their destinations within or outside the cell. Some proteins are further chemically modified at the Golgi body. The Golgi apparatus also produces lysosomes and cell walls. Lysosomes are single-membrane vesicles formed from the Golgi body. They contain hydrolytic enzymes. Lysosomes help to protect the organism from pathogens, or derive the food source of pathogens. Materials taken in by the cell are broken down by hydrolytic enzymes in the lysosomes. The products of digestion are then absorbed and assimilated by the cytoplasm. Also, the lysosomes help to break down worn-out organelles within a cell in a process called autophagy. When a cell is dead or damaged beyond repair, the lysosome releases its contents during autolysis. Ribosomes are made up of 2 subunits and each subunit comprises proteins and ribosomal ribonucleic acid. Ribosomes are either attached to the rough endoplasmic reticulum or suspended freely in the cytosol, which is the aqueous part of the cytoplasm. Usually, ribosomes are found in clusters. Ribosomes function as sites for protein synthesis. Centrioles exists as a pair of rod-like structures positioned 90 degrees to each other. They consist of 9 triplets of microtubules arranged in a ring formation. Microtubules are long hollow tubes which are made up of tubulins. Each triplet is attached to each other by fibril proteins. Centrioles do not have a clear role, since centrioles are absent in more complex plant cells. In some animal cells, they serve as microtubule organizing centers. Centrioles may also change the orientation of microtubules. Chloroplasts are bound by double membranes. The outer membrane is smooth and continuous while the inner membrane gives rise to membranes in the semi-fluid interior stroma. The stroma contains circular deoxyribonucleic acid and 70S ribosomes. Phoecoids are stacked to form grana, which are joined by the intergranal lamella. Chlorophyll, a photosynthetic pigment, are found on grana and intergranal lamella. Chloroplasts function as sites for photosynthesis. Sugars and other organic molecules are made in the process.
Thank you for watching. Don't forget to like and subscribe.